In this lecture, we're going to review the chromosomal abnormalities that can occur and also the diseases that result from these disorders. Following this lecture, you should be able to list and describe the various types of chromosomal structural anomalies, as well as the chromosomal number anomalies. You should also be able to define mosaicism, chimerism, and uniparental disomy, and compare and contrast the various diseases caused by chromosomal aberrations. So remember that we have chromosomal disorders, and these disorders are usually the result of either a loss, a gain, or an abnormal arrangement of one or more chromosomes. And these chromosomal disorders can cause excessive or deficient amounts of genetic material. The various changes that can occur in the chromosomal structure are inversion, duplication, deletion, microdeletion and microduplication, translocation, isochromosomes, ring chromosomes, and subtelomeric rearrangements. A deletion is a loss of a portion of DNA, and these can range in size from a single nucleotide to a larger portion of a chromosome to an entire chromosome. So the genetic material has not been transferred to another chromosome. A deletion is the result from two breaks that occur on a single arm, which leads to loss of material. So the loss of a long arm of chromosome 7 at band 28 of the long arm is written in a particular manner. And this is just an example of one type of deletion. So you would write this particular deletion as 46, X, X, DEL, which means deletion, parentheses 7, end parentheses, and then parentheses Q28. Parentheses. So these types of chromosomal disorders are written in a very particular way. A derivative is an abnormal chromosome formed from portions of two or more chromosomes. A duplication is the presence of an extra segment of DNA from the same chromosome. So it results in redundant copies of genes or redundant copies of gene portions. An inversion is the result of two breaks within a single chromosome. So instead of losing the intervening DNA, there's reattachment with a reversion of the reading frame. This inversion could be paracentric, which doesn't involve the centromere, or pericentric, which does involve the centromere. And again, how to write an inversion would be INV, which means inversion, parentheses 9, close parentheses, parentheses Q21, Q31. An isochromosome is the presence of only the short or long arm material on either side of the centromere. So for example, on chromosome 12 that only has short arm material, it would be I for isochromosome, parentheses 12p. A microdeletion or a microduplication is either the loss or gain of a portion of a chromosome that is considered small cytogenetically. However, it's large enough to affect multiple genes, and examples of microdeletions are Prader-Willi and Angelman syndromes. A ring chromosome results from a break in both arms of a chromosome that rejoin to form a ring. Usually you're going to get a loss or deletion of some material. So the example of how to write that would be little r, which means ring chromosome, and in parentheses you have your chromosome number and the locations at the short arm and the long arm. A subtelomeric rearrangement is common and thought to be one of the causes of mental retardation. The reason for this is it's found in up to 10% of children that have unexplained moderate to severe mental retardation. And what a subtelomeric rearrangement is the regions near the end of the chromosomes, which are the subtelomeric regions, have small deletions and duplications in them and can rearrange. A translocation is the physical rearrangement of two chromosomes where the material from both chromosomes are transferred from one to the other. It occurs when there's a break in two chromosomes and the material is traded between the two. Translocations can be inherited or they can just arise spontaneously. They can also be Robertsonian or reciprocal. And you can see their written designation. So this written form, the 46, XX, T for translocation, parentheses 922, parentheses Q34, Q11. So the first location, Q34, pertains to the break point on chromosome 9, and the second location, Q11, refers to the break point on chromosome 22. A Robertsonian translocation occurs when you have a break at the centromeric regions of two acrocentric chromosomes. When these join, you get a loss of minimal material on the short end of each centromere. So the loss is so small that you might not have a deleterious effect. The new chromosome is composed of the long arms of the two fused chromosomes, which results in a cell that only has 45 chromosomes as opposed to 46. So carriers of Robertsonian translocations are usually phenotypically normal. However, they can have offspring that might have an effective monosomy or trisomy. Reciprocal or balanced translocation happens when there's no net loss of DNA following a translocation. So carriers of reciprocal translocations are usually normal, but the offspring are at risk for having miscarriages and phenotypic abnormalities. So this is the schematic that we've seen in a previous lecture showing some of the chromosomal structural abnormalities. We already talked about a lot of these abnormalities, but we're just going to go into them in a little more detail and then talk about the diseases. So some changes in chromosomal number include aneuploidy, which is a gain or loss of one or more chromosomes, in a manner that the total number is not a multiple of 23. So two types of chromosomal number changes that are the result of aneuploidy are monosomy, which is the loss of a complete chromosome, or trisomy, which is the gain of a complete chromosome. We also have non-disjunction, where you have failure of chromosomes to separate, as well as polyploidy, where you have more than two complete sets of chromosomes. Other chromosomal abnormalities, and we already talked about these two, are mosaicism and chimerism. So remember, mosaicism is the presence of two population of cells with different genotypes in one individual that's developed from a single fertilized egg. And this can result from a mutation during development and is propagated to a subset of adult cells. And the most common mosaicisms involve trisomies. Chimerism is when you have two or more genotypes that arise from the fusion of more than one fertilized zygote, and this happens in the early stage of embryonal development. It's more common in animals than in humans, although it does occur in humans. So each population of cells keeps its own character, and you get a result of it results in a mixture of these cells in the tissue. So sometimes phenotypically you can see the two different cell types in the tissues. So here's a picture of some chimeras, and on the left-hand side you see the chimeric mouse, and it has two different colors in its fur with its offspring. And on the right is a chimera that was scientifically made, and it's called a geep, where they combined a goat and sheep cells. Uniparental disomy is when you have both copies of a chromosome or part of a chromosome or a gene that comes from just one parent. So this happens due to non-disjunction during meiosis. So during meiosis, one gamete gets both homologs and the other gamete gets neither. So the offspring is effectively homozygous for either the paternal or the maternal alleles. They don't have one from the father and one from the mother. Structural or numerical chromosomal anomalies are rarely inherited, although they can be. So germline cases are attributed to dysfunctional meiosis during gametogenesis. Somatic disorders usually arise at any point after fertilization. And you can detect chromosomal anomalies using cytogenetics, fluorescence in situ hybridization, or what's called FISH, or amplification techniques such as polymerase chain reaction, or PCR. Chromosomal breakage syndrome are usually caused by the expansion of a repeated sequence of 3 nucleotides, and that's called trinucleotide repeats. And these trinucleotide repeats can happen either in the coding or non-coding regions of a gene. So trinucleotide repeats are present in the normal gene, and the numbers of these repeats vary in length throughout the population. However, there's a certain number that if you go beyond that number of repeats, that gene becomes unstable. And so these extensions tend to expand more with each successive generation. And these chromosomal breakage syndromes may be due to defective repair of spontaneous chromosomal breakage. And remember, we talked about repair mechanisms in the previous lecture. So if you remember from previous lectures, the chromosomes are looked at by a method called karyotyping. And karyotypes of chromosomes are annotated in a very specific way. So they're expressed as the number of chromosomes per nucleus, and normal is always going to be 46, as well as the sex chromosome, where you're either going to have XX or XY, followed by any genetic abnormalities that are observed after karyotyping. So a normal karyotype would be 46, XX in a female, or 46, XY in a male. Annotations of genetic abnormalities include 46, XX, DEL parentheses 7 parentheses Q13. And this denotes a deletion, the DEL, in the long arm, which is Q, of chromosome 7 at region 13. The next annotation denotes a translocation between the short arms of chromosome 5 and 17 and region 13 band 3, you could see the 13.3, and region 13. The third example, 47, XX plus 21, is the karyotype for a female with Down syndrome, or trisomy 21. And this slide shows several of the descriptive abbreviations that are used in karyotype annotation, and you're going to see these in the next several slides when we're discussing the various diseases caused by chromosomal aberrations. So some of the disorders caused by chromosomal anomalies. You have your numerical disorder diseases, which include spontaneous abortus, Down syndrome, Edwards syndrome, Pateau syndrome, Turner syndrome, and Klinefelter syndrome. So spontaneous abortus. Approximately 50% of spontaneous miscarriage specimens, you can identify chromosomal abnormalities in these specimens. And only 0.5% of live-born infants have the same chromosomal abnormalities. So most chromosomal abnormalities are monosomy X45X, triploidy 69XXY, and trisomy 1647XX plus 16. And among live births, trisomy 21 or Down syndrome is the most common finding. So Down syndrome or trisomy 21, the karyotype of more than 95% of individuals with Down syndrome is trisomy 21. However, 4% have an unbalanced translocation and 1% are mosaics. The extra chromosome 21 is usually maternally derived, and it's the result of a meiotic disjunction in a normal female. So this female does not have an extra chromosome or anything like that. It affects 1 in 800 newborns, and the incidence increases with maternal age. So the incidence is 1 in 2,000 in mothers under 20 years of age. However, the incidence increases to 1 in 25 in mothers over 45 years of age. The symptoms of Down syndrome in neonates include hypotonia, hyperflexia, excess skin on the back of the neck, flat facial profile, as well as cardiac anomalies. Over time, you get additional features such as mental retardation, hearing loss, autoimmune thyroid disease, immunodeficiency, and leukemia. Almost all trisomy 21 individuals who survive beyond the age of 40 will develop neuropathologic findings similar to Alzheimer's disease. Another trisomy, trisomy 18 or Edwards syndrome, is a disorder where few individuals will survive beyond 1 year of age. The incidence of Edwards syndrome is 1 in 3,000 live births, and this also increases with maternal age, just like trisomy 21 does. Neonate features include clenched fists, intrauterine growth retardation, inguinal or umbilical hernias, cardiac defects, low-set ears, and rocker bottom feet. Beyond the neonatal stage, you get profound mental retardation. 50% of individuals with trisomy 18 die within the first week of life, and less than 10% will survive beyond 1 year. Pateau syndrome, or trisomy 13, is another trisomy disorder where you don't usually get survival beyond 1 year of age. The incidence is 1 in 5,000 live births, and again, trisomy 13 increases with maternal age. The features include polydactyly, microcephaly, sloping forehead, cleft lip and palate, retinal anomalies, abnormal ears, cardiac defects, genitourinary malformations, and over time you can get seizures, profound mental retardation, and deafness. 50% of individuals with trisomy 13 will die within the first month, and less than 30% will survive the first year of life. In Turner syndrome, you can actually get spontaneous abortions. However, those that are born alive tend to live through adulthood. Half of those born with Turner syndrome have a complete loss of one X chromosome, and the remainder have 45X mosaicism. There are three main karyotypes that are found in individuals with Turner syndrome. About 55 to 60% of individuals with Turner syndrome have the entire X chromosome missing, so they're 45X. 30% are mosaics, and the remainder have structural anomalies resulting in partial monosomy, where you can get a deletion of the small arm of the X chromosome, deletions of part of the short arm, or deletions or portions of both the small and the short and the long arms. In Turner syndrome, up to 10% will have some Y chromosomal material in their genome, and in these individuals there is some degree of masculinization. The incidence is 1 in 2,000 live female births, and neonates will present with lymphedema, a webbed neck, broad chest, widely spaced inverted nipples, short stature, and a low hairline on the back of the neck, as well as cardiac abnormalities. The lifespan for individuals with Turner syndrome that are born alive is normal, and ovaries that are normal in utero progressively get depleted of their ova, and by infancy there's no ova remaining. Klinefelter syndrome is present in 1 in 1,000 live births. In most cases are detected during childhood, but some are not detected until adulthood. Affected males tend to be tall, with long arms and legs, and have small testes. Some have mild retardation, and almost all men with Klinefelter syndrome will be infertile. Disorders that are caused by chromosomal deletions and microdeletions include Wolff-Hirschhorn syndrome, Cri-du-Chat syndrome, Angelman syndrome, Prader-Willi syndrome, Smith-Magenis syndrome, and Y chromosome microdeletion. In Wolff-Hirschhorn syndrome, you get a deletion of the short arm of chromosome 4. And affected individuals have what's called a Greek helmet face. They also have microcephaly, hypoplastic eye socket, cleft lip and cleft palate, a beaked nose, cardiac malformations, and mental retardation. Cri-du-Chat syndrome was named because of the characteristic cry that infants have with this syndrome. It's a result from the deletion of a short arm of chromosome 5, and some of the features include small stature, microcephaly, a moon-like face, a wide and flat nasal bridge, and mental retardation. In both Angelman and Prader-Willi syndromes, they're both the result of the same deletion on chromosome 15 with very, very different clinical manifestations. So in Prader-Willi syndrome, you'd get mental retardation, short stature, obesity, small hands and feet, hypogonadism, and the deletion is on the paternally derived chromosome 15. In Angelman syndrome, this is a deletion of the same band, but it's affected by the maternally derived chromosome 15. And Angelman syndrome causes mental retardation, ataxic gait, seizures, and inappropriate laughter. Smith-Magenis syndrome, these features are usually very subtle during infancy. Infants may be lethargic and might have some minor morphological anomalies. However, as these individuals grow, they display more prominent morphological anomalies, such as a prominent forehead and short stature. Also, mental retardation can develop, as well as visual problems, hearing loss, and sleep difficulties. Y chromosome microdeletion syndrome is caused by microscopic deletions in the Y chromosome, specifically within the YQ11, and this is the azoosperma factor region. This is the most frequent genetic cause of spermatogenic failure in azoospermia, which is no measurable levels of sperm in the semen. Other chromosomal disorders are trinucleotide repeat disorders, and there's numerous diseases that are caused from trinucleotide repeat disorders, but we're just going to discuss Fragile X syndrome and Friedreich ataxia. In Fragile X syndrome, the incidence is 1 in 2,000 male births, and it's the most common inherited form of mental retardation. Affected individuals also manifest autistic behavior as well as attention deficit and hyperactivity disorder. Physical findings include a large head size, prominent ears and prominent jaw, macroorchidism, which is large testicles, and tall stature. Female carriers are either normal or they may have mild mental retardation. In Fragile X syndrome, the underlying defect is found in the distal long arm of the X chromosome, and what you get is a series of CGG repeats, these trinucleotide repeats. Now, these CGG trinucleotide repeats are normally present at this location, which is the FRAXA locus. This locus is in close proximity to the FMR1, or familial mental retardation 1 gene. So normal individuals have 29 CGG repeats, but may have up to 200 CGG repeats. When that number exceeds 200 repeats, the FMR1 promoter becomes methylated, which is going to inactivate that gene. When less than 50 repeats are present, the locus remains stable. Once there's more than 50 repeats, the locus becomes unstable, which can cause an expansion. And the FRAXA locus, when it has between 52 and 200 repeats, that's considered a premutation. Carriers of premutations are phenotypically normal. If the locus has more than 200 repeats, that's considered a full mutation, and it's associated with phenotypic abnormalities. Friedreich ataxia is an autosomal recessive disorder. Affected individuals suffer from deterioration of the spinal dorsal root ganglia, and it manifests as gait ataxia or difficulty in walking, usually no later than age 20. Also, individuals develop decreased pain and temperature sensation, and some may have eye problems and hearing loss. It's caused by a GAA trinucleotide repeat expansion in the FRDA gene. So in fully affected individuals, they have more than 500 GAA trinucleotide repeats. Normal alleles are those with up to 65 repeats, and between 66 and 500 are premutations. Diseases that are associated with chromosomal abnormalities that cause short stature and dwarfism include achondroplasia, hypochondroplasia, and thanatophoric dysplasia. Achondroplasia is the most common form of dwarfism. The incidence is between 1 in 15,000 to 1 in 40,000, and the inheritance pattern is autosomal dominant. However, 80% of achondroplasia cases are sporadic and caused by a spontaneous mutation. Homozygosity is lethal, and individuals with achondroplasia demonstrate short stature with disproportionately short arms and legs, a large head, and abnormal facial features. Overall lifespan is normal, and the disorder is caused by mutations in the FGFR3 gene. More than 95% of these mutations are the result of a G to A mutation at position 1138. Others are from a mutation, a G to C mutation at the same position. Hypochondroplasia is characterized by short stature with disproportionately short arms and legs, broad, widened hands and feet, and macrocephaly. These features resemble achondroplasia but are usually milder, and they also result in mutations in the FGFR3 gene. However, these mutations are usually a C to A mutation at position 1620 or a C to G mutation at that same location. Both of these nucleotide mutations result in a lysine for asparagine substitution in the protein. In thanatophoric dysplasia, this condition is usually lethal in the neonate. Actually, thanatophoric means tending towards death. Features include bent femurs and a clover leaf skull deformity. Neonates usually die of respiratory insufficiency shortly after birth, and this disorder is also caused by mutations in the FGFR3 gene.